Okay, the second of two screencasts on the process of sugar metabolism or sugar breakdown, we're going to look at aerobic cellular respiration. So what happens after glycolysis? Um, so this is less meaningful when we're doing everything on the same day. But the mind's on, just what I'd like you to keep in mind here is which molecules store energy, oxidized or reduced. And we talked about this in the last screencast, so if you don't remember it, go back and rewatch that portion. From the picture below, there is no picture below. We'll do it in class. So the learning goal, we're learning to explain the chemical changes and energy changes that occur during pyruvate oxidation and Krebs cycle phases of cellular respiration. So the same as the learning goals were for glycolysis, just we're doing two different phases. The success criteria, you should be able to describe the events of pyruvate oxidation, the general important steps in the Krebs cycle. So similar to what we did with glycolysis, where we looked at things in overall phases, and you were aware of key intermediates. We're going to work the same way in this step. You should be able to explain the role and importance of both those phases to the overall process of cellular respiration, and you should be able to track the carbon atoms from glucose through those processes and the energy from glucose through those processes. Okay, so remember, our overall learning goal is the chemical changes and energy changes. So anaerobic processes do not require oxygen. Aerobic processes do. So the processes that we're going to learn about today are, or in this screencast, because you're probably watching them all on the same day, the processes we're going to learn about in this screencast are all oxygen requiring screencasts, or oxygen requiring processes. So keep in mind, when we left off, the bulk of the energy from glycolysis was stored in 2 3-carbon molecules of pyruvate, which I am going to represent by highly scientific little circles. Okay, so we have 2 of these 3-carbon molecules. The bulk of the energy from glucose is still in them. There's some energy in the molecules of ATP that we created. So we created a net of 2 molecules of ATP in glycolysis, and we created a net of 2 NADH molecules in glycolysis. So there's some energy stored in those, but all of the carbon and much of the energy from glucose is still found in these 2 3-carbon units. So what happens is the pyruvate is transported into the mitochondrial matrix, and it goes to an enzyme called pyruvate dehydrogenase, which is actually a complex of multiple copies of 3 different enzymes that all work together. And they take that pyruvate and convert it into a 2-carbon molecule, and they release a molecule of CO2. So what happens is 1 atom of carbon from each of those 2 carbon units is released as carbon dioxide. So it is now completely oxidized. And the remaining 2 carbons from each of those pyruvate molecules move into the next phase, into the Krebs cycle. Okay. Now, 2 lonely little carbons, they're teeny, they're weeny, they could get lost all by themselves in that big, scary mitochondria. So we actually attach them to a big, giant energy carrier, which I'm going to draw as a highly scientific sort of elongated octagon. And we call this molecule acetyl, or the whole complex is called acetyl this is why I wanted this put on my computer. It's cool. So I could do this on the active board. So the whole complex is called an acetyl-CoA. This big, huge carrier molecule is a coenzyme A. So think of it as the big kid that's walking the little kid to school, all right? The coenzyme A is a big molecule that's going to make sure these tiny little 2-carbon units don't get lost somewhere in the cell. So here's that shown graphically. The pyruvate is a 3-carbon molecule, and the pyruvate is going to get taken into the mitochondria. It's going to release a molecule of CO2. So one of its carbon atoms is going to get released here. The other 2 carbon atoms get fixed to this coenzyme A. And you can see here, and you'll start to see this pattern, it's the carboxylic acid groups that are going to get lopped off as carbon dioxide. So we've got this carboxylic acid on the end of our pyruvate or pyruvic acid molecule. That's going to get lopped off and released as CO2, and this 2-carbon unit is going to hang out. Now, because the pyruvate is being oxidized, this carbon is now completely oxidized. It can't have any more oxygen stuck to it. Because that pyruvate's being oxidized, we have to have a coupled reaction, right? So if something's being oxidized, something else has to be reduced. In this case, the molecule that's being reduced is that energy or electron carrier NAD+ that we talked about previously, and it's going to get converted into NADH. And we're going to see a lot of these NADHs made, and I'm going to talk a little bit more about what they do later in the note, and a lot more about what they do in class when we look at electron transport. So the remaining 2 carbon atoms, as I said, are an acetyl group, and that is attached to that carrier molecule, coenzyme A, and 1 molecule of NAD+ is used to produce an NADH. And the role of pyruvate oxidation is to prepare that pyruvate, the end process of glycolysis, and convert it into something that can begin the Krebs cycle. So it's that 2-carbon unit that's actually going to go to the Krebs cycle. And it does produce a molecule of NADH, which is going to be used to produce energy. So if you're doing an energy accounting, and we're going to do this all together in class, do an energy accounting of what we've got so far. We have 2 NADH being produced per glucose molecule from pyruvate oxidation, because remember, I lost my highly scientific diagram. We have 2 of those pyruvate molecules being produced per glucose, so 2 NADHs produced, 1 per each pyruvate. So the next is the Krebs cycle. It's also known as the TCA cycle or the citric acid cycle. The Krebs cycle is a cyclical pathway, thus cycle. And what we mean by a cyclical pathway is that we end up with the same thing we started with. It occurs in the matrix of the mitochondria. So my mitochondria, if you'll remember, it looks like, this is a highly scientific way of explaining things. I always think it looks a bit like a sausage with mustard. All right, so those like pretty beautiful sort of food-styled hot dogs where the mustard is a perfect little zigzag. All right, so remember the mitochondria has 2 membranes. It's got an outer membrane and it's got an inner folded membrane. The folds of these membrane are called cristae. The inside of this inner membrane, so the area I'm kind of shading in right now, is the matrix. Okay, so just a quick refresher of mitochondrial structure. This is the matrix. Welcome to the matrix. And this is the intermembrane space. So inter is between, so the intermembrane space, the matrix, and then obviously outside of the outer membrane is the cytosol. So carbons from glucose enter the cycle as acetyl-CoA. So they're going to move from the cytoplasm into the mitochondria, and they're going to get converted into acetyl-CoA through that pyruvate dehydrogenase enzyme, and they're going to enter the cycle as acetyl-CoA, leave the cycle completely broken down into carbon dioxide. And since the two molecules of acetyl-CoA enter the cycle for every glucose that starts the process of glycolysis, the Krebs cycle turns, in air quotes, twice for one glucose molecule. And this is really only going to be important in a context of doing some ATP math. So we'll take a look at an overview of the Krebs cycle quickly. And for those of you who watched the terribleness of my glycolysis screencast, you'll realize how much smoother this is going.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl.
Sorry, I'm going to pause briefly. The same as we were doing in the last screencast, and you've seen me doing so far, we're going to account for our carbons just as little circles because it's easier that way.
CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Okay, so again, we're going to focus on the broad phases of this cycle, so I don't expect you to memorize every single compound that's formed. So part of the reason why I like this animation is because it shows you things as 6-carbon molecule, 5-carbon molecule, 4-carbon molecule, few steps of rearranging to get a new 4-carbon molecule. So I'm really more concerned that you be able to track. Again, I can't stress this enough. Where is the energy at each step, and where are the carbons at each step? You don't necessarily have to memorize in each intervening or intermediate compound. All right. So the general phases of the Krebs cycle, as he mentioned, the Krebs cycle consists of 9 overall reactions, and we break it down into 5 sort of general phases. So the initial condensation, where we take that 2-carbon acetyl group and stick it onto something. An isomerization phase, oxidation, and that actually happens twice. There's an episode of substrate-level phosphorylation. So again, remember we talked about substrate is in an enzyme. So substrate-level phosphorylation is phosphorylation happening in an enzyme. And then oxidation, again, happening twice, and regeneration of that initial starting 4-carbon compound. So the first reaction is condensation. Acetyl-CoA delivers 1 2-carbon unit into the Krebs cycle. So remember, acetyl-CoA is like the big kid walking little kindergarten to where it needs to be. That CoA is really all it does is act as a really big molecule that's going to make sure that acetyl group doesn't get lost in the big old mitochondria. So it delivers the 2-carbon unit to the Krebs cycle. It reacts with a molecule of oxaloacetate to produce a molecule of citrate. So this is where it gets the name, the citric acid cycle, because the first product is citrate. And citrate is rearranged to produce isocitrate. It actually kind of all happens at the same time. Reactions 4 and 5 are oxidation. So the first oxidation steps convert isocitrate into a molecule of succinyl-CoA, and in the process, 2 molecules of carbon dioxide are released and 2 molecules of NAD+ are reduced. So again, we're going to talk about our coupled oxidation reduction reactions. If we have 2 molecules of carbon dioxide being oxidized, then 2 molecules of something have to be reduced. Remember that when something is reduced, it's becoming an energy storing molecule. So this NADH is going to store energy that we can cash in later in the cycle. All right. So if you go back to the animation and the way they represented it, I just want to show you this picture rather than fussing with my screencast on my pen. We've got our 6-carbon molecule. So basically what we're going to do is we're going to lop off a carbon dioxide. So this isocitrate is actually a carboxyl group floating around in the middle of nowhere. So we're going to lop off that carboxyl group. We get a resulting 5-carbon molecule, and we're going to lop off another carboxyl group and end up with 2 molecules of CO2 being there. And we've now produced 3 CO2s for every pyruvate that entered the mitochondria. So we've completely broken that pyruvate down into its 3 compounds. I'm actually going to play up to that point in the animation again for you.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced, and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs.
Okay, so we're going to stop at this. So we've got the second oxidation and decarboxylation. So far, from 1 molecule of pyruvate, we've made 1, 2, 3 molecules of NADH through pyruvate oxidation and the Krebs cycle. Now keep in mind, for every glucose, we actually have 2 of those pyruvate molecules made. So the next reaction is substrate-level phosphorylation. And again, substrate, enzyme. So this is phosphorylation happening in an enzyme. And I know I keep harping on this, but it's because we're going to look at an example of substrate-level phosphorylation, and we'll look at oxidative phosphorylation, but I can't really say anything to you right now other than that there's something called oxidative phosphorylation, because we're going to learn about that tomorrow. So ATP is produced by substrate-level phosphorylation, and in a substrate-level phosphorylation, you take that succinyl-CoA and you're going to remove the coenzyme A from that succinate group, or that succinate molecule, and you're going to replace it with a phosphate. That phosphate is then quickly transferred onto a molecule of guanine triphosphate. And for all intents and purposes, guanine triphosphate is energetically equivalent to ATP. So as soon as that GTP is made, we transfer that phosphate onto that molecule of, or onto a molecule of ADP to create ATP. The reason being is that ATP is used as cellular cache all over the cell, whereas GTP can't hook up with enzymes with proteins and do work the same way ATP can. So we just are taking this maybe less inconvenient form of cache and converting it to a more convenient form of cache. It's like if I'm going to the States on vacation, I'm going to take my Canadian money and I'm going to transfer it into American money. And at one point, it was virtually at par. So this would be the equivalent of me taking $1 of American money and cashing it into $1 of Canadian money because I can't spend American money in Canada. Reactions 7 to 9 involve 2 more oxidation reactions and the regeneration of that initial oxaloacetate that's required to start the cycle. So reactions 7 and 9 are coupled reactions. And what that means is that an energetically favorable reaction is used to pull forward a reaction that's maybe not quite as energetically favorable. And in the process, we reduce a molecule of FAD to FADH2 and NAD plus to make another molecule of NADH. FADH2 is almost the same thing as NADH in that it's a large energy carrying molecule or electron carrying molecule, and that's going to become important tomorrow. I feel like I keep repeating over and over again that it's going to be important tomorrow, but I am not lying to you. So as this happens, the reactions regenerate the oxaloacetate needed to begin the cycle. So the compounds I'd like you to know from this cycle really are citric acid and oxaloacetate. You should kind of have some idea of what succinyl-CoA is, but that's really the ones that I'm mostly concerned about. And again, it's because oxaloacetate will become an important compound when we get into photosynthesis. So at this point, we've completely broken down the glucose into carbon dioxide. Actually, by the time we get to that 5th step, we've completely broken down the glucose into carbon dioxide. So we can also say that glucose has been completely oxidized. We've added so many oxygens to each of the carbons in that glucose molecule that they can't have any more oxygen bound to it. And there's very little energy stored in those CO double bonds compared to what was stored in a glucose. And the way I always remember this, although it's a bit of an oversimplification, is that we can burn glucose. Things that are highly reduced, like methane is super reduced carbon. It's got 4 H's stuck to it. I can burn methane. I can burn glucose. I can get energy out of them in that way. I can't burn CO2. In fact, CO2 does the opposite of burn. It puts out a fire. So it's hard for me to get the energy out of that CO bond. There isn't a lot of energy stored in it. So I'm actually going to go back before I go to here and we'll watch the end of this animation again.
Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Okay, so just to finish up, take a look at the energy diagram from the beginning of the unit. So this was in the very first screencast in this unit I had you watch, was the very last slide, I think. Again, I said the main goal is you be able to track through all of these processes. Where are the carbons from glucose? Where is the energy from glucose? So after the glycolysis phase, the energy from glucose is primarily found in those two pyruvate molecules. So they have a little bit less energy than glucose did, but not a ton. Some of the energy is also found in the one molecule of NADH and in the ATP made in glycolysis. The glucose is all found, or the carbons from the glucose are all found in the pyruvate. Then you go through pyruvate oxidation. After pyruvate oxidation, the bulk of the energy is still found in those two acetyl groups stuck to the acetyl-CoA. Some of the enzyme or energy is found in the NADH that's produced in that phase as well. The carbons, four, I guess, if you count that there's two acetyl-CoAs made. So two of the carbons are found in the acetyl-CoA, and one carbon, so 2 carbons per glucose, are released as carbon dioxide after this phase. So carbons here, all in pyruvate. Carbons here are in acetyl-CoA or carbon dioxide. Then they go through the Krebs cycle. And when they go through the Krebs cycle, now all of our carbons from glucose are completely broken down into carbon dioxide. So we've made 6 molecules of carbon dioxide per glucose. Most of the energy here is now stored in those energy carrying molecules, the NADH and the FADH2. Okay? So the NADH that you made here, the NADH that you made here, the NADH you made up here, that's where the bulk of glucose's energy is trapped. So after those 3 steps, we've only made, I think it's 6 molecules of ATP per glucose through substrate-level phosphorylation. The bulk of the energy from glucose has been transferred into the NADH and FADH2 molecules that are produced in those 3 cycles. So right now, we're about here on that diagram, but all of our molecules are trapped. All of our energy is trapped in those molecules, and we've got to find some way to get it out. So tomorrow, or in this particular year that I'm recording this, Tuesday, we're going to look at how the energy that's stored in the NADH2, NADH and FADH2 is converted into ATP. So what I'd like you to think about over the weekend is the difference between stocks and bonds and cash. All right, so think about where you can use cash, where you use stocks and bonds, how you can get the money value out of cash versus how you can get the money value out of stocks and bonds. And that's going to help you to understand why the cell's making all these energy carrying molecules.